All right, thank you, Prof. Kabuka and Prof. Han. Our next speaker is Sir David Klenerman, who will speak on the next generation DNA sequencing. The Q&A segment will be moderated by Prof. Patrick Tan. Sir Klenerman, please. Thank you.
Thank you very much. So what I'm going to do is tell you a little bit about my background and introduce to you single molecule fluorescence and super resolution microscopy. I'm going to briefly show you how we are developing and applying these methods to study protein aggregation. Then I'm largely going to focus on DNA sequencing. And sort of the themes of the message are how new physical methods can enable new science. And following on what was just been discussed, there's a lot of luck in doing science. So you just have to take advantage of that if it happens. So this is me when I got my degree at Cambridge, and then I stayed on with Ian Smith and did a PhD. And this is my lab. So I have all this gas handling equipment, so the glass that I built myself. This big vessel here where I pump the gas through, and an infrared spectrometer. And the idea of the experiments was to detect the infrared emission from these gas phase reactions. So, for example, I was going to look at hydrogen atoms interacting with ozone to form AH and O2. So all the gas got, I formed the radicals up here. I have this big chamber where I collected the light, and it went over to the infrared spectrometer. I then spent a couple of years at Stanford trying to do high overtone chemistry, and then I returned and I worked in industry at British Petroleum for 7 years. So here's me in the lab. I have these lasers, and the idea of these experiments was to do nonlinear laser spectroscopy to try and work out how corrosion inhibitors work and lubricants worked. And in 1994, I came back to Cambridge. And one of the big advances in physical chemistry at the time was the ability to detect single molecules. And that's really what I focused on since I arrived back in Cambridge. And this works by exciting a fluorophore with a laser, with light. And the fluorophore can emit fluorescence at a different color, at a shorter wavelength. And the main idea behind single molecule fluorescence is you can excite the fluorophore again and again and again, typically 1 million to 10 million times, and that's enough photons to easily detect it with a modern detector. So typically you work at low concentrations, typically picomolar, and only have one molecule present at the time. And importantly, as you'll hear in this talk, this is really the ultimate in miniaturization if you're doing experiments with single molecules. So just to calibrate everyone, this is a length scale. So here's a bacterium, a cell, a mitochondria, and there's a fundamental limit, the diffraction limit, of about 200 nanometers. So if I have a virus or a protein or small molecule which is smaller than this, it's going to show up, if I use fluorescence, as a spot of about 200 nanometers in size. So this is just to show you what our labs look like. It's not a great video, but it's just to make the point that we build our own instruments. They're under computer control. They're on very stable tables. We have a bank of lasers that we can control the illumination. And eventually, we scan out. We have an inverted microscope coming soon, hopefully. We have an inverted microscope where we put the sample. Here it is. And then we have a very sensitive detector here. So this is what the lamp looks like, very colorful. We relay the light, and the light, it comes up an objective, undergoes total internal reflection. Here we've got a T cell. We've labeled the T cell receptor with an antibody, which has a dion. And this is what we see. So these individual T cell receptors diffusing around on the cell surface. We're interested in understanding how the T cell is so sensitive and selective at identifying a cancerous cell. The other big revolution in my field, I'm sure Stefan Hell will talk a lot more about, is his ability to beat the diffraction limit. So this is just an example of an object that's blurred by diffraction. But what you can do is illuminate single molecules. So single molecules are still blurred by diffraction, but I know I have a single molecule, so it must be at the center of this spot. And so typically I can localize where this molecule is with about 20 nanometer precision. So I replace this blurred spot with this higher precision spot. And then I can, if I do this again and again and again, I can build up a super resolution image of the object of interest. So one of the problems we're interested in is neurodegenerative disease. So in this, a small soluble protein such as alpha-synuclein aggregates and it ultimately produces these Lewy bodies. But it's known that it's these small soluble aggregates that are formed on the way to producing these Lewy bodies are toxic. So we've got a big effort in developing methods to detect these small soluble aggregates and work out how they're toxic, and particularly focusing on doing this in the species in humans. So the experiment that we discovered worked was we took cerebral spinal fluid. So this is the fluid that bathes your brain. You can get it from a lumbar puncture. We took cerebral spinal fluid from someone who's got Parkinson's disease and put it on our microscope with a dye called thioflavin T. So thioflavin T is non-planar in solution, but it can become planar and highly fluorescent when it binds to the beta sheet structure in these aggregates. And much to our amazement, we could see these small bright spots in our image. Each of these image spots is a single aggregate. We think of alpha-synuclein. And it raised a lot of questions. How do these aggregates change as someone develops Parkinson's disease? Are these aggregates actually alpha-synuclein? And so we've managed to super resolve these aggregates. So this is a 100 nanometer scale bar. These are the aggregates we see in cerebral spinal fluid. We see a lot more and bigger and rounder aggregates in serum. And we see a high proportion of these big aggregates in people with Parkinson's disease. So we think this is potentially a diagnostic for Parkinson's disease, and we think this is a general method. We think we can use this, for example, for the American football players or the rugby players who get these bangs on the head and then get likely to get early onset dementia. So that's just a sort of brief introduction to my background and the type of work I do. So I'm now going to focus on the rest of the talk on next generation DNA sequencing. So DNA is in almost all our cells, and it's a code consisting of 4 bases: adenine, guanine, cytosine, and thymine. And the order of these bases contains all the information for building and maintaining an organism. And A is always paired with T, and G is always paired with C, and this enables a cell to synthesize a new strand of DNA. So again, C with G and T with A. So it's fundamental to life on Earth. And if you want to sequence DNA, you require a way of decoding the sequence of DNA. You need a sequencing technology. So why do you want to sequence DNA? You really want to understand life. And we differ in bases in 1 in 1,000 between all of us. And you want to understand these differences. You want to be able to understand why people are more likely to get disease. You want to be able to detect and diagnose disease. You want to be able to intervene before people actually get diseases. And you want to monitor the effectiveness of treatments. But the problem is that there are 3 billion bases in the human genome. So the way this was done previously was Sanger sequencing. I'll go through this very quickly. You have your DNA, you have a template primer, and you put in nucleotides, and you have nucleotides that are blocked, that are labeled red, green, yellow, and blue. So what you do is the polymerase makes millions of copies of the DNA. It puts in the nucleotides according to the code, but occasionally one's blocked by one of these terminators. And so you end up with all these different lengths of DNA, and you run them down a gel. The small ones come off first, the large ones come off later, and you use a laser to detect the fluorescence of the labeled nucleotide, and hence you can work out the sequence. So you have one tube, and you can detect about 500 bases per run. So it's $3 billion to sequence the first human genome, and this was the average sequence. And then to resequence individual human genomes at the time, this is back in the mid-90s, it was about 30 to 50 million. And what people wanted was a way of sequencing the human genome for $1,000 or less. So back in 1995, we weren't actually thinking about how to sequence DNA. We were trying to do this experiment, which I show here, with 2 postdocs. We have a template primer of DNA, and we have, this is the polymerase, and these are the nucleotides. And we were trying to use single molecule fluorescence to watch the polymerase putting these nucleotides into the growing strand of DNA in real time. And back in 1997, this is me, this is my colleague Shankar. We didn't get this experiment to work very well, and so we went to this pub, it's very close to the chemistry department, to discuss how we might get our experiment to work. And what we did when we had these discussions was come up with this way of doing DNA sequencing. So this was the idea we came up with. We have our template primer, we're going to have a polymerase, and we're going to have our 4 bases color-coded, and the polymerase is going to put in the right base, in this case an A. And these bases are going to be designed so we only put a single nucleotide in, so we can read the color of the incorporated base, in this case it's an A. We're then going to remove the fluorescence and undo the termination, and then repeat the cycle again and again and again. And the idea was to extract the DNA, fragment it and attach it to the surface, so we got single molecules on the surface. As long as these are, say, a micron apart, we can resolve them individually. So we essentially have millions of sequencing lanes on the surface, and we're going to sequence everything in massively parallel fashion. So we're going to get all these short reads of about 30 bases. We have the average human genome, and in this way we can reassemble the whole human genome. And this is a calculation I did back in 1997. So for me, if there was a eureka moment, this was a eureka moment on a flight to Germany when I did this calculation in my head and realized that we could sequence very rapidly. So there are 3 times 10 to the 9 bases in the human genome. I'm going to assume we have a 300 by 300 array of these single molecules of DNA. I'm going to assume it takes 10 seconds to read the fluorescence. So I have 10 to the 5 bases divided by 10. So I can sequence about 10 to the 4 bases per second. So one machine is going to take about 100 hours or 5 days to sequence a human genome. So this, we realized, was a pathway to actually solve the problem and be able to sequence human genomes at very low cost. So we debated what we should do, and we were eventually convinced that we needed to form a company because we needed increasing amounts of money in order to actually solve all the technical problems involved in developing the sequencing technology. So we came up with formed a company called Celexa. It doesn't actually mean anything. It's a sort of play on single molecule, it's a single mole, and Shankar thought any good company like Xerox should have an X in it, so we put an X in it. This is our, one time we were going to use these DNA hairpins, so this is it in the logo. So we formed this company. It started off with just 2 postdocs working on this problem in our chemistry department. This is the nucleotides that Shankar designed. So we have a fluorophore with a cleavable linker and a reversible terminator on the nucleotide. So the company worked on this problem for about 10 years, solving a series of technical problems. And the only difference from what I've outlined to you was, instead of studying single molecules on the surface, we used a solid phase amplification to convert each single molecule into a little cluster of about 100 molecules. And this was technology developed by Pascal Meyer. So what you see when you do these experiments are these random dots on the surface, each color-coded for the nucleotide that's been incorporated. So if you look at here, you can see yellow, green, blue, and red. So this is our first instrument. You can see our logo here, and that in 2006 could sequence 1 billion bases per run. So just want, so in 1997 we had the idea. By forming a company 10 years later, we've got an instrument that's actually up and running, and we're selling to people. In 2021, the instruments can do 6 trillion bases per run. So in 1997, it was 10 to the 5 bases per instrument. In 2021, 10 to the 13 bases per instrument, an improvement of a factor about 10 to the 8. Number of sequencing lanes went from 100 to 10 to the 10, again an improvement of a factor of 10 to 8. And probably the most important thing, the cost has gone down from $1 billion to $200, so a reduction in cost of a factor of about $1 million. So we went from this situation, this is a picture, front cover of Science, some sort of cocktail party and one insect talking to another, well, have you been sequenced yet? In 2009, we published the first sequence of an Afro-American taken on our technology using our method. So in the UK, there's been a big effort to sequence a large number of the population, 100,000 genomes, and follow people through their life to see how much their genetic variation contributes to the disease they develop. I think there's a similar project here in Singapore. So in 2001, I think there was only been one sequence, and as you can see, this is a logarithmic scale. The number of whole human genomes has gone up, so up at over 1 million genomes. This isn't quite up to date, over 5 million genomes in 2017. The cost is done enormously, so we're now, in 2001, again, this isn't quite up to date, we're now down at about $200 per genome. So I just wanted to talk you through some examples of using this technology now, just to show you what's possible. So the first example, we have someone with lung cancer. We're going to sequence the tumor, we're going to sequence the normal lung, and we're going to compare the sequences. We're going to assemble these short reads and look for the differences. In this example, there was a mutation in this RB1 gene, which seemed to be causing the cellular proliferation. And in many cases, there's drugs that will act on this, and you can come up with a treatment plan for the patient. The second example is, again, cancer. What you can find is small amounts of DNA circulating in the blood, and you can sequence, if you sequence it at sufficient depth, you can detect this small amount of circulating DNA. And again, you can come up with a therapy that targets the cancer. So there's a big effort in using this technology in cancer. The next example is in rare genetic disease. About 1 in 17 people get rare genetic diseases. In this example, it's a child who presents at 5 months with developmental regression and seizures. And there's rapid whole genome sequencing of the child and the mother and the father. And again, you look for the genetic differences. So this is the number of transcripts that are different. But these are triaged. They have to be functional. They have to cause a disease. And it has to be deleterious. And so if you do this, you end up determining that this novel homozygous variant, ATP7A, that causes disrupted copper metabolism. The doctor could work out that the child had Menkes disease, and there's a treatment for this, so the child could go home. And in the UK, this is now being made available to newborn babies who have undiagnosable disease. This is another example. This is a gene test on this baby here. Discovered that they had they had a benign cancer and they didn't need any treatment, so the baby could go home. This is another example of a child who was sequenced and could be treated and could go home. So for me, this was actually quite an eye-opener. I originally thought we were developing an instrument to empower other scientists to sequence DNA. So I was very proud, actually, when I heard about this on our local news station, that they were doing this to children at Addenbrooke's. And it really shows you, for me, that we went from this calculation in 1997, 20 years on, it's actually being used to save people's lives. The next example is COVID. As I'm sure you're aware, that we've been sequencing the virus. This is, again, a logarithmic scale. So sequencing the virus So this goes up to 100,000, a million sequences of the virus, looking for viruses, changes in virus sequence and different strains of virus. So it's been widely used for survey and to detect changes in the pathogens. So really I just wanted to finish there and just reflect on this. So really I think what I want to first highlight is science as a social activity. So I'm a physical chemist, Shankar is a chemical biologist, and it's only by us working together that we could actually come up with this idea. And in the company, we then had engineers, people who done computation, people who could mutate the DNA polymerase, and it required a large number of talented people really to get this to work. I just wanted to highlight it's hard work. Like the discussion earlier, these experiments often do not work, and you have to pick yourself up and try again. But you have the advantage that you're doing something that no one's really done before, and so by definition, you're going to come up with problems, and it's fantastic when it works. So Shankar and myself had no idea that this was going to work, and we had no idea it was going to work as well as it's turned out to work. So the only thing I think that we did well was actually try the experiment. So we actually organized things so we can actually see if our idea worked. It could have failed for all sorts of reasons. We couldn't solve some surface attachment problems. It could have failed because someone came up with a better way of doing DNA sequencing. But the thing that, if I had my time again, was very important that we actually tried. So I think the main message for the young people in the audience is you're not going to know if something's going to work or not unless you try. And you can enhance your chances of doing something that might be significant by always working at the frontiers of the fields. So my advice is work at the frontiers and try experiments and get used to them failing, because they're going to fail if you're working at the frontier. So you may only be at the right place at the right time once, and you should recognize that and act on that opportunity. So the biggest thing that happened to me in my scientific career was coming up with the way of doing DNA sequencing and actually forming the company to do that. I just want to highlight for the funders that the idea came from basic curiosity-driven research. Neither Shankar and myself were working on DNA sequencing. We were doing basic research, trying to understand how single molecules work and watching biology in action, and that's where the idea came from. And I just highlights, I think, how new chemistry, and particularly from my background, new ways of measuring things can solve major societal problems and improve people's lives. Thank you very much.
Thank you, Tony. Thank you. Hello, can you hear me? Testing, great. So my name is Patrick Tan, and it's my great honor and privilege to be able to moderate this session. Maybe to provide an input, the institute that I lead, the Genome Institute of Singapore, is 400 people that are all focused upon different ways of using the instruments that David has invented. And it's just, I dare say that this is a foundational technology that has really revolutionized all aspects of biology. I can't think of a single aspect that even Dr. Koboka's previous lecture alluded to the fact that it's using DNA-based barcodes, and that's being dissected using Illumina sequencing. So obviously, with many questions from the floor and also online, but maybe I can have the prerogative to start off. Let's talk about the philosophy of science. And I was attracted in your bio sketch about this ability to have people from different disciplines collide together and to think of new solutions. What is it about the environment that you think is important for that, in order to nurture that sort of interdisciplinary, and for how do you get, it doesn't happen by chance, I think, or what makes good science?
So I think a lot of it's personal. So Shankar and myself had arrived back in Cambridge to start our lectureships at the same time.
Yes.
So we're both young scientists looking to do something interesting. And we realized that by working together, we could do something that was more interesting than working individually. But you also have to get on with each other, so we also get on very well. We were put in touch by one of my colleagues. So in Cambridge, we're organized in colleges, and I think the colleges are a good way of going across disciplines. So Chris Abel, who's an organic chemist, put me in touch with Shankar and suggested we try and collaborate. I mean, Cambridge chemistry, until 20 years ago, the physical chemistry was entirely separate from the organic chemistry. So we're in silos. So I think breaking down the silos is very important. And the collegiate system is a way of going across disciplines.
Fantastic. I think questions, Sir Hunt.
Yeah, this was absolutely fascinating because you know, I've never had a successful company, and nor a really good idea the way you did. But I'm curious, I wish you'd said a bit more about the difficulties that you faced during the development of the technology. And I suppose one question one could ask was, you know, what was the most serious problem that you had to overcome in the actualization of the machines?
I think it was a series of problems. Once we solved one problem, we moved on to the next problem. So there was nothing.
But there were lots.
There were lots, and so the first thing was being able to put one nucleotide in it and visualize it. That was the first problem. But the nature of the process is if we can do one nucleotide, you know, we just have to be able to do that
Over and over again.
over and over again and improve the efficiency. So it was really, getting it to work, which was done in the chemistry department, and then getting it to work better and better. And, you know, one of the pivotal things, like I mentioned, was that there was the last piece of the jigsaw was going from a single molecule to these little clusters.
Yeah.
But it really did take we had 170 people working in the company, and it needed 170 people to solve those problems. So it's not possible in the sort of small 10, 20 groups that you have in a...
Yeah, so how did you manage 170 people?
I didn't.
Somebody else.
So one of the pivotal questions was, did we want to go out with the company? Did we want to be CSO of the company? And both Shankar and myself decided that this didn't play to our strengths. We were better at coming up with the ideas and doing academic science. And so they brought in someone from pharma who was experienced in managing 200 people. And the quality of the people and the quality of the management all really contributed to getting it working. So I wouldn't know how to manage 200 people.
But you stayed associated with it?
Yeah, so the way it worked is we were on the board of the company and consultants for the company and spending 1 or 2 days a week at the start, but it did decline over time. So we were very actively involved right from the start. And the company was taken over by Illumina in 2007, and Illumina continued to engineer and improve the technology.
Thanks very much.
So we'll take a number of questions. I will say that Sir David actually will be speaking on Thursday also at an event organized by SG Innovate. So I think that if anybody that wants to find out more, particularly about the company, please attend that event. We'll take a few questions from the floor, and then we'll go on to the online. So maybe can I have the next question? If there's anybody on this side, please wave your hands. But a question for you.
Hello, thank you very much for this lovely talk. I'm a bioinformatician, so my job makes sense to people like you who created these lovely inventions. I analyze sequencing data. And of course, this motivates me a lot. So I have two questions, two motivational questions. The first one would be, what are your tips for people who want to start a company from scientific projects?
When do you want to start a company?
So, I mean, for us, our experience of forming a company is a little bit different from most people's, because we literally went to the VCs with an idea written down on a piece of paper with no proof of concept. So they didn't quite know what to do with us, because this was particularly something that clearly, if it worked, it was going to be very valuable, but we had nothing to show that it worked. So to their credit, that they eventually decided to fund it. But I guess my advice would be that it's a team effort. The VCs are there on your side if they decide to fund you, and you should go and speak to them. They explained to us that even if we didn't, they already had a DNA sequencing company, they would then put us in touch with another VC who might fund the work. And they're very connected. So my advice is to go and talk to a VC, and they'll give you feedback, even if it's not they don't think it's a commercial proposition at the time. And I also, my other bit of advice is talk to other people who've set up companies, because it's a different way of thinking about science. It has to work. You have to get to the end. You can't just do something to see what it works. But on the other side is you have access to much larger amounts of money than you can as an academic. So it's really important, I think, you talk to people who've done it previously who can give you good advice.
And what about team-wise? What would be the key things to consider when choosing the right team to work with?
What makes a right team?
So I think what we did well is we got the best people in for every single position. So I think it's all about getting quality of people in. It's just quality, and don't take anyone if you've got second thoughts. It's all about getting high quality people in and focusing them around a problem and managing that well. I guess that would be my advice.
Thank you. And the final question is, of course, we all have ups and downs in our research. We are all very young. And you gave lovely tips at the end of your presentation, but if you could go back to your younger self, what would you say? Apart from the idea, of course.
I mean, I don't think
Advice.
I would change anything. I think you, as a young person, you just have to get used to things not working. You just have to get used to experiments not working and find a way of picking yourself up, have another hobby, something you can distract yourself, and just, and either it would eventually work, or you'll think of a way around it, or you'll stop for a year or so and come back to it, you know, if it's still an important problem. But there are ways around it, but I think it's, the nature of research is you're doing something no one's done before. So it's unrealistic to expect it to work. First time, fifth time, it's going to take a lot of effort.
I hope our funders hear, our research funders should hear that. Maybe we'll take another question from the microphone, and then we'll go on to some of the online questions. So please, next one.
My name is Leo Zeitler. Thank you very much for your lovely talk. It was very interesting. I have a rather technical question. So I'm working with DNA repair, so a dynamic process. We use different NGS data for, well, comparing what is going on during that process. But something that I just realize is that it is very important to make sure that a certain protein that I take a look at with one particular type of NGS data would line up with another type of protein in another experiment, right? So this rather technical question would then be, is there a possible way that you can imagine how we can identify always the same cell? What I mean by that is can you imagine a method where we can barcode a DNA of a particular cell so that we can later, with our NGS data, line up different data types, for example, for different proteins, or also, like in my case, for damage?
Barcoding cells. So,
I mean, there are single-cell sequencing methods now. I'm not sure if they're amenable to your problem or not, but you know, potentially you can just, certainly what we're working on is coming in with a fine pipette, sucking all the DNA out and then sequencing it. So I think that type of technology, I mean, there are also ways of sequencing in getting the data in situ. So there are ways that people have been working on to address your problem, I think. It's not something we've done very much on, but I think there are ways now to get spatial information.
Yeah,
Thanks.
thank you very much. I was just interested whether you are aware of the, like, combining these two benefits of single-cell sequencing and having, like, sequencing of a bulk of cells altogether.
So I think there are ways of improving single-cell sequencing. I mean, we certainly work with a nanopipette, and the idea is just to extract directly the DNA from that cell. And so we'll know exactly where it comes from and then sequence. But we need to get that working.
Cool, terrific. Thank you very much.
Maybe just a point is that on the basis of the technologies like Celexa, a whole suite of new companies have grown up around that technology, simply finding new ways to feed it into the instrument. And I think this is really a whole ecosystem that's really started from your
exactly.
work.
I think the community have really, I mean, it's all fine to give this talk, but, you know, Illumina, then other scientists have really made it what it is today. We just started the stone rolling down the hill.
Yeah, and it's gathering momentum every day. We'll take some questions from online first to give due credit to our online audience. I don't know if they're going to come up on the slide, but maybe the first one in getting back to the science now. What do you see are the new emergers, the new challenges that are facing genomics now? I mean, that remain to be tackled for the next generation. So from you alluded to the problems of Sanger, then you had short read sequencing, and what's the future of this technology? And for scientists here, what should they think about as the things to do, the hard things that remain to be done?
I mean, I think, in my mind, it's largely application, sequencing enough people so that we really can understand how much your gene sequence predisposes you to get disease, and starting to use that medically. I mean, if we're now done at $200, I'm not sure it changes very much if it's $100, and it's hard to, I think that if there was a technology that reduced it by an order of magnitude down to $10, I still think that would be valuable. But you know, even storing the information would probably cost you.
The compute is going to be.
So I think in my mind, it's all about application, developing tools to actually interpret the information and acting on it. I think that's where I think the direction of travel. I think there'll be a tipping point where we start to sequence newborn babies because it's much more cost effective to have their DNA sequence and know which diseases they're predisposed to and treat them early. So I think that's going to happen in the next 10 or 20 years.
Fantastic. So the $10 genome is the next challenge for everybody in the audience. Another question from the floor?
Hello, everyone. So I am Alisa Lola, a PhD student from Japan. And before I proceed with my question, I would like to say that I just came from a failure with my experiments and relating to sequencing. And my supervisor told me to use NGS, but the problem is it costs a lot, especially for third world countries. So I'm actually from the Philippines. And in third world countries, this is actually a dream technology for us. So my question is there a possibility that this technique will be lesser when it comes to its cost, and because it's really out of reach, I believe, for most of the third world countries, even though we really wanted to do it?
I'm not sure. The cost is now $200 a genome. I think it's slowly coming down. I mean, there are alternative technologies like Oxford Nanopore, which is potentially cheaper. It depends what you want to do. So Oxford Nanopore, so if people know it's a pore and you pull the DNA through and you measure the current blocking and can distinguish between the bases based on the current blocking. But the problem with the Oxford Nanopore is accuracy. So I think it's fine if you wanted to identify different bacteria from their sequence. But for human genetics, I think that the lack of accuracy is still a problem for them. So there's Oxford Nanopore, which I think might be potentially cheaper for you if you can deal with the reduced accuracy. otherwise, I think it's plateaued. I think the cost of the dyes, for instance, that we use, it sort of means that there's a level that the cost can't really go below very much. So it's hard to see it going down much more than where it is now, not more than a factor of 2. So if you need an order of magnitude, you probably need another method, or Oxford Nanopore need to improve their accuracy.
Yeah.
Thank you very much.
Thank you. I'm going to take a question, which is maybe as a spokesperson that really started this whole enterprise, and it's from online, and it refers to some perceptions in the community that DNA is deterministic. You know, if you're born with a particular, that's it. Your fate is charted for the rest of your life. So the question based on genetics, do you believe that if one of the parents is a psychopath, would a child inherit? I know it's a bit of a stretch, but maybe you can comment on the role of DNA in as you mentioned, being the blueprint of life. But is that really all there is? And in terms of determining health and disease, maybe you can comment on that.
I work a lot on Alzheimer's and Parkinson's. I think for Parkinson's disease, it's 30% genetic and the rest of it's environmental. I think for Alzheimer's, it's higher, maybe it's 50%. So there's the genetics, and then there's the environment. I think there's a very interesting question around mental health and how much your genetics contributes to that. That's really been understudied. I know there's been a study on anorexia, but I think we're scratching the surface with mental health. But no, I think that there's probably quite a lot of disease that's environmental and not genetic. I mean, before we started, or when we started, there was this big debate about how much would be genetic and how much would be environmental, and how similar or different diseases will be for individuals. So now I think for a lot of diseases, it's maybe half genetic and half environmental. And certainly for Alzheimer's, the idea is at least you can control the environment, you can eat well and exercise and stay healthy. That's the bit you can control. So your life is not determined by your genome.
That's very good. Important. It's influenced, but your fate is still your own. Maybe another question from the floor.
Good afternoon, everyone. I am Awais Qasim from Pakistan, a Biosecurity Champions Fellow. So, being a Biosecurity Fellow, I am concerned with the dark side of this technology. Could it be misused? And if it is misused, for example, how can we prevent the adverse effect of this technology in future?
I think this is a question for society, how we use the information. So I think when James Watson started the Human Genome Project, they had people looking at the ethics of this. You know, whether you can make a decision for a newborn baby based on their genome. I think that the feeling was you couldn't do that. You had to certainly wait till people reach adulthood. So definitely, I mean, it's like a lot of science, technology, it's, you know, or like fire. You know, fire you can use to cook your food and stay warm, or you can use it to burn things down. So that's a decision for society. So far, I think we've acted well, and it's been used properly. And I think it's less the sequencing; it's more the genetic editing where there's potentially more moral issues. But definitely, it's a societal problem. We have to decide as a society how we want to use this information. And so far, I think, you know, it's been done well, but we need to continue to watch this, particularly if genetic editing starts to be more widely used.
Yes, like a lot of sequences of polioviruses were published, and it was argued that it comes under the umbrella of dual-use research of concern and evidence in the history. So there was a lot of discussion on it.
Yeah.
So it was all about like next generation sequencing, because it was sequenced and then the sequence was published, and it was argued that it can be used to construct these viruses by some invaders or some other person who intentionally misuse it.
So again, I think this is a problem for society. I mean, I think the sequencing of these viruses and bacteria, you didn't need our technology. I think this is generally a question of responsible publishing and being a responsible scientist. So I don't know the answer that, I don't have a good answer to that. I think we all have to act responsibly, and if we think we're publishing the sequence of a virus that could be used for biological warfare, then we need to maybe think again about publishing that.
Yeah. Thank you.
Thanks. So we'll take one last question and then we'll call it. But maybe I can highlight this. And yeah, as you alluded to this, in the days now of CRISPR engineering, don't you think that we will now have the ability, the technical ability to edit human genomes in the near future or distant future? And what do you think that will mean for human health and disease? I mean, when do we cross the line between fixing things versus modifying? It's a difficult question, but maybe I'd like to get your thoughts on that.
I mean, I still think there's a long way to go with CRISPR editing. I think, you know, you as likely to give person of cancer as to cure. But again, I think once these tools have become available, it's a question for society. If it was safe, I think there's a big issue determining if these things are safe. And then, you know, it's a question for society, what diseases would we feel it's acceptable to modify? And you know, and what diseases is it unacceptable? I mean, I think that there's a book I've read that discusses this, it imagines this situation. Again, there's this film Gattaca, I don't know if people have seen it, which imagines a world where people are valued based on their genetic sequence. So I think these are discussions for society. I don't have any real insights on it.
But perhaps that is the challenge when the science becomes so so pervasive, or it does really reflect challenges that society now has to grapple with. And perhaps that's the biggest triumph of some scientific developments when it becomes a question of ethics. I think we have our time is up, and please join me in welcoming Sir David. I think it's a wonderful presentation. Thank you.
Thank you very much, Sir David and Prof. Tan, for the engaging presentation and discussions. So this concludes our morning's plenary sessions, but do also know that we have a lunchtime online engagement with the Earth Observatory of Singapore, EOS, and it'll be on promoting safer and more sustainable societies. This will commence 12:30, so it kind of already commence on the Give Me platform. But now we also invite you to join our speakers outside for lunch, and we encourage you to reconvene back here by 1:25 p.m. for our afternoon sessions. Thank you and see you soon.